Hello everyone, and welcome to the second session of this intermediate three-part ARSET training titled Solar-Induced Fluorescence, or SIF, Observations for Assessing Vegetation Changes Related to Floods, Droughts, and Fire Impacts. I'm Dr. Erica Podest, a scientist at NASA's Jet Propulsion Laboratory and also an instructor with the ARSET program. Today's session will provide an overview of OCO2 and OCO3 observing modes and SIF observations, followed by a demonstration on using the snapshot area mode data from OCO3. We have two invited experts, Dr. Yunji Liu and Jackie Ryan, both from NASA's Jet Propulsion Laboratory. This is the training outline. There are 3 sessions associated with this training. Today is session number 2, and it consists of an overview of OCO2 and OCO3 observing modes as well as SIF observations. There will be a theoretical section and a demonstration on how to access and visualize OCO3 SAM mode data. The third session will be next Wednesday, October 29th at the same time. There is a homework associated with this training, and that homework will open on October 29th. So during the last session, we'll open up the homework, and it's going to be due on November 12th. You can access the homework through the training webpage, and participants who have attended all 3 sessions, live sessions, and complete the homework by the due date will be given a certificate of completion. And just a reminder, these are the prerequisites for this training, the fundamentals of remote sensing, and a couple of years ago we did an introductory training on SIF. It was SIF and LiDAR to assess vegetation change and vulnerability, so the prerequisite would be a review of the SIF component of that training. In the next couple of slides, I'll provide an overview of today's session. So today's trainers are myself. I'm acting as the ARSET training coordinator for this webinar series. And we have 2 invited experts. We have Dr. Yunji Liu. She's a scientist at NASA's Jet Propulsion Laboratory. She will provide the theoretical component of today's webinar. And we have Jackie Ryan. She's a data visualization developer at the NASA's Jet Propulsion Laboratory as well, and she will be leading the demo. Here are the objectives for today's session. By the end of this session, participants will be able to identify the characteristics and operating modes of OCO2, OCO3, and other SIF datasets, recognize the synergistic use of SIF with other datasets, recognize some of the applications of SIF, recognize how the OCO3 snapshot area mode or SAM mode can be visualized using an open source Jupyter notebook as a means to evaluate land change due to fire impacts, run this Jupyter notebook environment from lesson 1, and run the OCO3 SAM notebook to analyze and visualize vegetation change due to fire across 2 case study sites. And finally, quantify the SIF-GPP relationship by comparing remote sensing data with OCO3, remote sensing data from OCO3 with eddy flux tower data at the 2 case study sites. How to ask questions. Please write your questions in the questions box, which is located in the bottom right. There are 3 points. There's a menu that will pop up. Select the first option, which is Q&A, and there you can write your question. Feel free to enter your questions during the presentation, and we will try to respond all of the questions during the Q&A session after this presentation. The remainder of the questions, if we don't get to answer all of the questions, We will answer them in the Q&A document. So we compile all of the questions into a Q&A document, and we will post that document on the training webpage in about a week. So today's session is focused on OCO2 and OCO3 observing modes and SIF observations. Our guest instructor is Dr. Yunji Liu. As mentioned earlier, she is a principal scientist at NASA's Jet Propulsion Laboratory, and she is an expert on SIF. So we're very grateful to have her lead this part of the training. Thank you very much, Dr. Liu, and welcome.
Thanks, Eric, for the introduction, and thank you all for joining the second part of the ARSET training on SIF. In this part of the lecture, I will first give an overview of the Orbiting Carbon Observatory 2 and OCO3 observing modes and their solar-induced chlorophyll fluorescence SIF observations, and then followed with a brief introduction to the level 3 SIF products. In the end, I will talk about application examples using these observations. So OCO2 and OCO3 both observes two main observables, column CO2 and the SIF observations using reflected sunlight, but they are in different platforms. So here the animation shows the column CO2 from both OCO2 and OCO3. And OCO2 is on A train, so it has pole-to-pole coverage depending on the seasons, and it has fixed like 1:30 p.m. equatorial crossing time, and was launched in July 2014. So it has more than 10 years of record. So it had the footprint size is pretty small. It's about 1.3 by 2.3 kilometers, but it has a very narrow swath. So it only had 8 footprints across track. It has about 16-day repeat cycle. But OCO3 is mounted on International Space Station, so it only has observations within 52 degrees latitude bands. It also has different coverage depending on the season. And the observations also have different time of the day overpassing time. So this is very unique compared to OCO2. So it has irregular repeat cycle and also irregular overpassing time. It was launched in May 2019. So it has a little bit more than 5 years of record. So here I will talk a little bit about observing modes. OCO2 has 3 observing modes. So nadir, glint, and target mode. For the nadir mode on the left panel, it's looking straight down. So it provides the highest spatial resolution footprint size on the surface. So it has better coverage over like partially cloudy or like the surface with high topography. But nadir observations doesn't have very good signal-to-noise ratio over dark ocean surface, just because ocean is highly absorbing for sunlight at nadir mode. So this comes to the glint mode. In the glint mode, the spacecraft points the instrument toward a bright glint spot. So the solar radiation is reflected from the surface. So this increases the observation the signal-to-noise ratio over the high latitude and also over the ocean surface. So all over the ocean surface, we have glint observations. And then for the target mode, the observatory locks its view onto a specific surface location and then retain that view while flying overboard. So the target mode are primarily used for validation purpose. Besides the land nadir, line glint, and nadir observations, OCO3 has this very unique snapshot area maps. For the SAM observations, OCO3 scanned this 80 kilometer by 80 kilometer area within 2 minutes. So it has a little bit broader observation coverage compared to target modes. And this plot showing the spatial distributions of SAM observations focused on the SIF. The yellow location shows the location with a little bit with lower SIF lower SIF values, representing lower productivity, while the purple sites are the high SIF sites. And then you can also request additional SIF observations by filling up online form on OCO3 SAM website. So I put the link for both the form and OCO3 SAM website here, and between the request time and the first collection, it takes about 16 days. Here are two examples of OCO3 SAM SIF observations. The left is over Cairo, Egypt, and the right over Incheon, China. So both sites are semi-arid region with crop coverage. So you can see that the crop and forest area has much higher SIF value, indicating much higher productivity compared to the background. So for both sites, you can see that feature. These are two examples of SIF maps from the OCO3 website, where it has all the column CO2 and SIF maps from the SAM locations. So the left shows the SIF maps over Hubble forest flux tower sites in the wintertime, while the right is at the same location but during the summertime. So you can see during the wintertime, the SIF value are much lower because of lower productivity, while the right has much higher SIF values. And I can also see this spatial gradient. So it has much higher values over the top right corner, indicating higher productivities of the plants. So this animation displays monthly mean SIF values from OCO2. It highlights this consistently high productivity throughout the year in tropical regions, as well as this seasonal shift you can see over the mid and high latitude. So during the boreal summer, the Midwest shows very high productivity, primarily due to crop growth. And you can see that over the high latitude, there is no observations during the winter, just because OCO2 using reflect sunlight and there is no light, so there is no observation during the winter time. Well, this animation presents a biweekly SIF observations from OCO2 and OCO3 together. Well, the SIF spatial patterns are very similar to the one that I just showed, but the distinct sampling pattern of OCO2, OCO3 are much more obvious here. You can see like the OCO3 has this irregular spatial coverage, but OCO2 had this fixed pole-to-pole observation coverage. As mentioned earlier, so OCO3 is mounted on the International Space Station, so it has this irregular orbit and also irregular local overpassing time. And this allows OCO3 to observe SIF at different time of the day across its different overpasses. So these 6 plots display the local overpass time measured at the difference between the local passing time and the local solar noon over a 60-day period. So this data shows that OCO3 local overpassing time can range from approximately like -6 to 6 relative to noon time. So because of this different overpassing time, the OCO3 observations can help us understand diurnal cycles of plants' productivity. This shows an example of a SIF diurnal cycle observed by OCO3. This is over Santa Rita, California. Although the observations at different times of the day were collected across multiple overpasses, but the pattern generally shows a higher productivity during the noon and early afternoon, while lower productivity during the early morning and the late afternoon. And this example shows regraded target SIF observations. During target mode, the different footprints has very large overlaps. So we can take advantage of this feature and regrade the target mode observation to a much higher spatial resolution than the original footprint size. So this regraded product is at 500 by 500 meter resolution instead of the original 1.3 by 2.3 kilometer resolution. And this is over Caltech site in Los Angeles. On the left is the SIF value in October 19th, while the right shows the SIF value in April 2022. For California, in October, we have much little water available, so the productivity is very low, shown here as low SIF values, while during the spring, we have much more water available, so that has higher productivity and shown here as higher SIF values. And this is the spatial distributions of the target sites across the globe. And those sites are also corresponding to surface remote sensing TCON sites, which are used for validation of the CO2 observation from OCO2 and OCO3. So those sites are mainly concentrated over North America and Europe, and some sites over East Asia. Both OCO2 and OCO3 have very narrow swath, as I mentioned earlier. So this results in a very significant spatial gaps between observation tracks. So this can become very inconvenient for a lot of data users. So in this next section, I will talk about the level 3 gap-filled SIF products that derived from OCO2, OCO3 level 2 observations. Unlike level 2 data products, level 3 had this wall-to-wall spatial coverage and also has much higher temporal coverage. This schematic plot illustrates a general process that has been used to generate level 3 SIF products. Different approaches generally just combine the satellite-derived like vegetation indices such as EVI, fraction of PAR, or meteorological variables such as air temperature, vapor pressure deficit, soil moisture, and then combined with the machine learning techniques to derive a relationship between the input variables and the output variable SIF, and then we can use this trained model to predict the SIF values where we don't have SIF observations. There are a few level 3 SIF products that's based on the OCO2 level 2 SIF. So GoSIF is one of the most broadly used products. It's generated by University of New Hampshire. So it has 3 temporal resolution, 8-day, monthly, and annually. It has 0.05 spatial resolution. It also has GPU products corresponding to these resolutions. And SIF was produced by University of Columbia. It has 4-day temporal resolution and same spatial resolution as GoSIF. Well, OCO2 005 products is generated by Cornell University. It has biweekly temporal resolution and same spatial resolution as GoSIF and SIF. And this shows example of OCO2 level 3 product and also corresponding level 2 product. So we can see the difference between the level 2 and the level 3 product. On the top shows annual mean SIF at 1 degree resolution, and the right shows maximum SIF also at 1 degree resolution, while at the bottom 2 panels shows the corresponding level 3 products at 0.05 resolution. So you can see like for the level 2 SIF products still have some temporal gaps, still has some spatial gaps, while for the level 3 products, it has this very nice wall-to-wall coverage, but retains this spatial characteristics at the level 2 products. For example, both shows the maximum SIF values over the Midwest during this in the maximum SIF maps. For level 3, for OCO3, as I mentioned before, that because OCO3 is on International Space Station, so it has different time of the day at different overpassing time. And this actually allows the community to generate diurnal level 3 SIF products. So the left panel shows the hours from local noon for these 3 days, while this animation shows the hourly level 3 SIF products derived from OCO3 SIF. So it has showing this very nice diurnal cycles of OCO3 of the SIF values. And this data also available on this link. This shows example how the level 3 diurnal SIF products can help us understand the diurnal cycles of GPP over the globe. So the plot shows the difference between GPP in the afternoon and in the morning for 2 July months. The top shows the dry July months, while for the bottom shows wet July, and the wet and dry are defined at each pixel. And we can see for either dry July or wet July, the afternoon GPP is lower than the morning GPP, indicating that the productivity is reduced in the afternoon due to the water stress, while in the dry July, the depression due to this water stress is even larger, which is very obvious in the southeast of US and also over the tropical region, indicating that the plants is much more water stressed during the dry year in July. So this indicates that level 3 diurnal SIF products can help us understand the diurnal cycle of the plant's photosynthesis. Another level 3 diurnal SIF products I want to introduce here is called GeoSIF. It combines OCO3 SIF and the greenness from a geostationary satellite called GK2A. So GK2A is situated over Eastern Asia and Oceania region. It has reflectance and shortwave radiation and also vapor pressure deficit derived from this satellite. GeoSIF has hourly temporal resolution and at 2 kilometer spatial resolution. And the top left shows the OCO3 SIF values, and the top right shows the reflectance, the greenness products from GK2A, while the bottom two panels shows the level 3 SIF in the morning and the level 3 SIF in the afternoon. So it shows the morning SIF has much higher values than the afternoon SIF. This shows the similar feature as I just showed in the previous slide, that the afternoon productivity is reduced just because of the water stress, especially during the summertime. The next, I will talk a little bit about synergistic use of OCO3 SIF and eco-stress data. On the International Space Station, it has a suite of the satellite missions that observe the Earth. The eco-stress is one of those satellites that observes the surface temperature and also retrieves water stress. I will give a brief introduction of the eco-stress instrument. So the eco-stress has spatial resolution about 70 meters and by 70 meters. So it has much higher spatial resolution than OCO3, but it also has very wide swath. So swath width is close to 400 kilometers. The plot on the right shows OCO3 SIF overlaid on top of the land surface temperature retrieved from eco-stress. You can see that the higher SIF values are on top of the lower temperature, This is because when plants photosynthesize, it releases water to the atmosphere and at the same time absorbing heat, so then cool the land surface. So this can really tell us how effective the plants converts water to productivity. So the community had defined a quantity to measure this. It's called water use efficiency. It is defined as the ratio between the productivity. Here we can use SIF to represent productivity, and the evapotranspiration, which is the amount of water released during photosynthesis, which can be derived from land surface temperature from ecostress. And OCO3 project team has generated this co-located ecostress and OCO3 data products, and it's available on NASA data service. And also I provide the link here. Here I show an example of using both SIF and ET from ecostress to investigate the diurnal patterns of vegetation photosynthesis and the coupling between the water cycle and the carbon cycle. And the top two rows shows the SIF diurnal patterns and ET's diurnal patterns over Amazon. So you can see that both SIF and the evapotranspiration increases toward the noon time and decrease toward the afternoon time. That's because we have more sunlight, so you have more productivity, but at the same time you release more water. But for water use efficiency, is actually decreasing close to the noon time. That means the plant is more water stressed close to the noon time, and also means it needs more water to fix the same amount of carbon at noon relative to the morning time. So this example tells us that we can use SIF and ET together to understand the coupling between the water cycle and the carbon cycle at diurnal scale. At last, I will give you a few examples of using SIF to understand the climate impact on the productivity and how we use SIF to improve the crop yield predictions. The first example investigates the response of the productivity to climate anomalies. Here is showing the response of this productivity to 2020 heat wave in Siberia. And the top panel shows the temperature anomalies in March, April, May in 2020. And it shows that the temperature anomaly can be above 5 degrees Celsius in this region. And in year 2020, Siberia region experienced the warmest spring in the record. But corresponding to this warm temperature anomalies, we can see this increase of productivity. This is based on the GoSIF GPP products. So this indicating the warmer temperature in the high latitude in springtime, it actually promotes a plant's growth. This is because over the high latitude, where it's normally very cold, so the plant's growth is limited by temperature. So when the temperature is warmer, the plants actually grow better. So this example tells us that we can use SIF products and also the GP products to understand the climate anomalies on the plant's growth. The second example I want to show is how we can use SIF and GBP products derived from OCO2 and OCO3 to understand the drought impact. So this study looked at the drought impact on the photosynthesis over Southeast Asia. So the left panel shows the land cover types over this region. The green color represents forest, while the yellow represent the crop, mainly cropland. The middle panel shows the drought index averaged between September 2014 and August 2016, about 2-year time period. So red color means much more severe drought. Relative to this, corresponding to this severe drought, especially over the central part of this map, with the GOSIF shows a large reduction in the productivity, especially over this cropland area. So this example indicates that GOSIF products, either the SIF derived level 3 products, could help us understand the drought impact on the productivity. So the last example I want to show is how we use SIF to improve the crop yield predictions. As we all know, it's very critical to know the crop productivity for early warning of the food insecurity or for the economic planning purpose. Well, the crop yield is normally generated by the statistical method at the end of the harvest season, which can be very late for the planning purpose. Well, this study demonstrates that we can use SIF to generate low latency crop yield prediction. So the left panel shows the crop yield prediction based on the climate data and also SIF, OCO2 SIF data. And the middle panel shows crop yield prediction over Midwest US, only use climate and land surface temperature, while the red panel shows the observation from statistical method. So the comparison of these three plots, you can see that the SIF-based prediction better captures the spatial distribution of the observation than the prediction only based on the climate. So this really shows the promise of generating the low latency crop yield prediction based on SIF products from OCO2. So this comes to the summary and some resources that you can check later on. So OCO2 had this global observation coverage, while OCO3 has observation at different times of the day that are really complementary. So we have both level 2 and the level 3 SIF products available, and this can be used to understand the impact of climate perturbations on vegetation health, and can also be used in drought monitoring and crop yield predictions. And here I list a few websites that I have talked about in the lecture, including OCO3 website and also the products that I have mentioned throughout the slides. With that, I will hand it over to Jackie. Thank you so much.
Thank you, Junjie. Now we'll begin the second exercise of this SIF training course covering the OCO3 snapshot area map mode and flux tower data by looking at case studies in 2 different regions of the United States. This is following up from our previous session introducing SIF data and how to work with it. If you've been following along from the first exercise, you should have already installed the course materials and have the Jupyter Notebook environment set up. If you have not done so yet, please review the README in the code repository or the instructions that we provided in part 1 before proceeding with this exercise. The first thing I'm going to do is open up my Jupyter Notebook in my browser. And so I'll turn over to my desktop so that I can do that. And you can see I already have the Jupyter Notebook code open in my text editor, although you can use the terminal as well if you prefer. And I'm going to run the setup script that we ran in part 1 again. And there's no harm in running this script more than once. And when I do so, it's going to open the Jupyter Notebook in my browser. And we can see now I have the Jupyter server running. So I'll pull my browser window down to my screen. And here you can see our notebook exercise 1. And I'm going to turn over to the second exercise. With our Jupyter environment open, we'll want to make sure that we're looking at the Notebook Exercise 2, OCO3 Snapshot Area Map, as you can see on my screen. So now that we have that out of the way, what is SAM mode? While OCO2 and OCO3 share the same spectrometer design, OCO3 incorporates a new component, the Pointing Mirror Assembly, or PMA. This hardware addition enables the instrument to rapidly sweep a mirror back and forth inside the optics, expanding the observation swath. Within time windows of several minutes, OCO3 can collect data over a nearly contiguous 80-kilometer by 80-kilometer region. This capability makes SAM particularly valuable for SIF measurements, as it captures fine-scale vegetation heterogeneity that would otherwise be missed due to the spacecraft's long revisit times. In this exercise, we'll discuss what SAM data looks like, how to retrieve it, and improve upon the method that we used for plotting granules from the first exercise to incorporate footprint coordinates for each sample. Once we have a handle on plotting SAM SIF data itself, we can talk about how SAMs are acquired repeatedly at different sites over time to build up a picture of vegetation dynamics in different seasons. The function of SAM observations is similar to that of eddy covariance flux towers, a well-established ground-based measurement technique that is used by ecologists to study vegetation dynamics. We can compare data from these two sources to determine if we're getting the same story, so to speak, from the data in both cases. To accomplish that, we'll correlate gross primary production, in other words, GPP, with OCO3 SIF values from SAM mode. The first site that we'll be using as a case study in the exercise will be the University of Michigan Biological Station in northern Michigan in the United States. This site will provide a baseline for ideal conditions when studying a temperate deciduous broadleaf forest, and we'll contrast our results here with the second site that'll be mentioned later. The site lies in a rural area on the northern tip of Michigan's Lower Peninsula and is comprised of about 60% vegetation cover with some agricultural fields and small towns interspersed in the surrounding area. If you want to investigate a particular site on your own, one of the easiest ways to do that is actually with Google Maps. So I'll actually go to Google Maps so that we can pull up this site ourselves. And what I'll do is I can either input the coordinates of the site itself, or I can go to the rough location if I know where that is. In my case, I'll be inputting the coordinates of the site itself. I've included those coordinates in our fourth notebook that we'll be discussing later. For now, we'll skip a specific discussion of that. So here's the latitude and longitude coordinates of our site. And I'll talk about where I got that information later. But first, I just want to discuss what this location looks like. And so I've put this into Google Maps, and you can see that if I switch to the satellite layer, this is the location of the case study site that we'll be discussing today. When I zoom out, you can see that this location is on the Lower Peninsula of Michigan. Here's the Upper Peninsula. And when I zoom in to the highest zoom level, the highest resolution data that we have available, you can see that there's an interesting feature here, which is the tower itself. It looks somewhat similar to a cell phone tower. The next thing we'll do to investigate this site is turn over to the OCO3 SAM website itself to investigate this tower location in the mission repository. And the website that we use for that is included in the introduction paragraph of our notebook. So I'll go to this link here, which you can see the URL is up here. And this website contains a map of all of the various sites that OCO3 has studied using the SAM mode over the life of the mission. You can find a specific SAM site by heading to the curated SAM webpage that we're looking at right now and searching for it by name or finding it on the map. The different colors for these pins represent different types, in other words, different local ecological conditions that can guide our expectations of what level of plant activity we can find at that location. Let's look at our University of Michigan Biological Station site just by zooming in on the map and looking around for the location specifically to see how that site is categorized. So if I click on this pin, you can see that this is the site that we're interested in. It's called SIF, University of Michigan, USA, and the OCO3 database, or SIF UMB is its short name. The type of the SIF site is SIF high. And so the classification that we've given this particular site is that it's a location where we can expect to find high SIF values during the growing season. The nearby site that we see to the north over Sault Ste. Marie with a blue square represents a fossil site. And this is a location where we would expect to find industrial emissions, more urbanized area, and increased emissions, perhaps lower SIF compared to a rural or wilderness area like we have in the case study site that we're using. You'll also notice that there are yellow pins and squares representing SIF low sites, such as this one to the west. And this is where we would expect to find less dense vegetation and ecosystems like grassland or rural or wilderness areas. So let's turn back to the info bubble for the site that we're interested in, and we'll use that site name that was mentioned up here to actually search for the SAM acquisitions that were acquired at this site. And don't worry, we'll actually be plotting what these SAM acquisitions look like so you can see the actual data that we're talking about. But first we need to find what data we can actually retrieve. You can see when you click on the pin down below in this form, the site name gets populated for you automatically. And that site name should be sufficient to find the SAM acquisitions. So if we just type search, we can see a table with all of the SAM acquisitions at this particular site. The table of results describes the time of each observation as well as the number of soundings, which roughly corresponds to the number of samples we can expect to find in the data from each SAM acquisition. So here's our column with soundings, and you can see some of them have a value of 0 and some of them have a much higher value. To choose the best possible example, we should therefore select a SAM acquisition with a large number of soundings to ensure that we find one with lots of sample points to look at. There are a couple to choose from in just this first page of results, so I'll choose the August 11th, 2019 example for no particular reason other than that it has a large number of soundings. When we click the plots button on the right side of the table, we can see that the OCO3 team has already processed the SAM acquisition with a number of different products. So I'll click that now. The first product that we're presented with is the XCO2 data, which is the primary product of the OCO3 mission. However, when we scroll down, we can also see that there are a number of other different pieces of information, including a SIF 757 nanometer product. And this is the one that we'll be reproducing today. There are two important details we should note before heading to the notebook to actually reproduce that plot, though. Number one, we'll want to note down the geographic extents of the plot data, which are helpfully marked along the X and Y axes. The extent of SAM coverage will vary slightly between acquisitions at the same site. So you will need to adjust these values a little bit for each date you want to visualize, or else use a larger bounding box. The other important detail to note down about the plot is the large red star on the map. And this marker represents the location of the tower site associated with the region. And recall that that tower site is the location that we were looking at in Google Maps. So let's look at this plot a little closer so you can see the exact details that I'm talking about. Remember, we'll want to note down this bounding box, the extent in latitude and longitude, as well as the location of our red star marker on the plot. Okay, with all of that out of the way, we can finally turn over to the notebook and begin running some code. As with the first notebook, in the first cell, we'll just import some necessary dependencies that we need. So I'll run that now. And with that out of the way, you can see I've also included the same information that I just discussed in a static form in the explainer text for the notebook in this section. In the second cell, we will supply the data of interest and the dataset, which is the same OCO3 SIF dataset that we used in exercise 1. So again, we're using the JESDISC downloader that we used from section 1. And we'll get the granule for August 11th. Now that we found the granule, we need to filter the data before plotting. To do this, we'll pull up the measurement mode and quality flag variables from the granule. And we know to do this by looking at section 4.7 of the OCO3 Data User's Guide. I won't pull up the User's Guide for this particular exercise, but you can look at it on your own if you're interested. Measurement mode 3 is referred to as an area map, and this is what we're actually going to be filtering the data on. We'll also filter to just select points with a quality flag of 0 or 1, which, if you recall, are points that have a quality of best or good. Additionally, the last filter I'm using is the SAM extent variable that you can see here in the code cell itself. And this extent is based on the latitude and longitude extent that I asked you to note down in the SAM plot from the OCO3 website. So now that we've selected the actual filter criteria that we want to use for creating our plot, the last thing to do is to run the cell itself. So an important detail is that we're using the same plotting function that we use for plotting any other SIF granule from the first exercise. In this case, we get a plot that looks like this. Now, this plot is pretty okay. It looks somewhat similar to the SAM plot that's on the OCO3 website. Similar enough that we can tell that we're looking at the same data. So if I scroll down, I've included the SAM plot itself, and you can see that the shape of data points and the values that we're using, or the values that we're observing, are pretty much the same between the two, with some minor cosmetic differences. So that tells us that we're at least retrieving the correct data. On the other hand, one of the most crucial differences you'll notice right away is that the samples in the official plot are represented by quadrilaterals at varying angles. So let me scroll back down to that so that you can see it. And here we see the quadrilaterals representing each of the SIF samples. One of the variables provided in the granule file that we haven't considered yet are footprint vertices. Each sample acquired by the instrument has an associated geographic extent of about 2 kilometers cross-track and 1 kilometer along track. When using SAM mode, the samples or pixels of the data become skewed as the PMA moves, adjusting the view zenith angle as it goes. To characterize SIF observations as accurately as possible, we should incorporate this information into our plot so that we know the exact area covered by each SIF value that we see. This is especially important in regions with a lot of spatial heterogeneity, such as where the transition between urban and forested areas is particularly sharp. One other piece of information we'll incorporate in our improved plot will be the tower site coordinate. I've written a function called plotSam that is included in this cell. So this next cell contains the function plotSam. And this will actually incorporate the footprint coordinates to make our plot from up here look more like the official plot down here. Specifically, the way that we'll do that is we'll use the geolocation footprint latitude vertices variable and the same one for longitude. And we'll also use the matplotlib patches module to create a set of polygons containing these footprint vertices as the vertices of the quadrilaterals used to represent each sample. And don't think too hard about that. We're just taking the list of these coordinates and putting it into this patches.polygon class that you can see here. Since these patches will be in the same order as the SIF sample values from the granule, we can color map each polygon with its associated value using the set array method in our patch collection that we've created up here. And this will automatically handle assigning the correct value to the correct quadrilateral. Another point that we'll add is satellite imagery base map using the contextually module. For the scale of this plot, I've used a zoom level of 9, which I think strikes a good balance between the detail of the satellite data versus the amount of data that's required to actually display the plot. Each zoom level is about 4 times higher resolution as the previous one, 2 times in each dimension. So if you have a slower internet connection or you want to use less data, you can choose a zoom level of 7 or 8, depending on your specific needs. And that comes at the trade-off of lower resolution for the base map. The last detail that we'll want to note down is that our original plot used an equirectangular projection. For the updated plot, we're using Web Mercator to match the projection of the official plot. And you'll notice the difference there in terms of the subtle difference in the shape of the data itself. So here you can see that the angle of this tail of SAM samples is somewhat different than the angle of the same feature in the official plot. And that's due to projection differences. So accounting for all of those cosmetic differences, as well as the very important inclusion of the footprint vertices, let's see how our own version of the official SAM plot looks. So I'm running the cell. It's going to take about 30 seconds. And that's because we're actually downloading the satellite imagery to use for our base map. And here we can see that the code has completed. And our result looks very close to the official SAM plot. Despite this, the important detail is that we are now incorporating the shape of the SAM footprints into our visualization. A single SAM plot on its own can be useful for context on a specific day and to get a sense of the SIF behavior in your region of interest. But we can only truly build up a picture of vegetation dynamics around a site through many successive observations across seasons and years. So let's move to the second section now, where we'll be performing a more long-term analysis of our UMBS case study site. Now we'll compare the SAM acquisitions to the flux tower data collected by instruments located at that red star on our plot. At ecovariance flux towers measure carbon dioxide exchange between ecosystems in the atmosphere. Since plants take in carbon dioxide and release oxygen when performing photosynthesis, we can measure this behavior by tracking minute changes in gas concentrations around the tower site. The towers are also effectively weather stations, and the additional data they collect can be used to differentiate the motion of gases in the environment due to wind and other effects from the influence of primary production. Although some towers measure SIF, this is somewhat uncommon. So we can't perform a direct SIF to SIF comparison between the tower and OCO3 for this particular site. When we look at tower data, the two variables we're most interested in are net ecosystem exchange, NEE, and GPP, gross primary production. In this exercise, we'll pull GPP values from the UMBS tower. Data from towers all across North America are archived on the Ameriflux website and can be accessed for free with an account. In this demonstration, however, we're not going to guide you through creating an account or downloading the data yourself, since you can do this on your own later. That also means that we haven't automated this step, so you'll have to manually download data for any particular site that you want to study outside of our two case study sites. As I mentioned, we've already downloaded the tower's data and included it in the CSV files in the same directory as the notebooks themselves, which you can see over here. Let's briefly pull up the Ameriflux page for the UMBS tower anyways, so that we can see what the information that gets provided for a given tower looks like. And I'll do that by clicking on the link that we've included in the description text. Okay, so looking at the Ameriflux website, we can see that the coordinate that I input into Google Maps at the beginning of this lesson is included right here. We've also got a description of the vegetation information around the site, which I also mentioned at the beginning of this exercise. A helpful picture showing context of what the tower and the surrounding area looks like. And we have links to download the data down here. Ameriflux data is provided in a CSV format at monthly, daily, and even sub-daily timescales of either 1 hour or every half hour. For our purposes, using daily tower data will be sufficient. And going back over to our notebook, this is the CSV file that we've provided in the course materials itself. And here you can see that file name in its full detail. Let's talk about the file name itself so that you can get a sense of what we're actually working with. So for a given CSV file with Ameriflux data, AMF tells us that this is Ameriflux data. USUMB tells us that the site identifier code is for the UMBS tower that we're interested in. FluxNet refers to FluxNet data, as opposed to BADIM, base, or any other type of data set that might be included for this tower. The subset identifier tells us that this is only a subset of the variables. However, since we're only interested in NEE and GPP, that's sufficient for our use case. DD tells us that this is the daily time cadence data. And 2019 to 2021 tells us that this data only covers the years 2019 to 2021. I've manually cropped the number of dates in this data to include data only from the lifetime of the OCO3 mission. If you had downloaded this data yourself, you would get 2007 through 2021 worth of data. So let's take a look at what we're actually going to be doing in this code block. We'll open the CSV with the GPP data and create a time series plot of its values. For comparison, we'll also plot the average SIF value from the area around the tower site, which has already been pre-processed in the JSON file that you can see down here in the included course materials. The code to perform this step is included in the optional appendix notebook that I briefly looked at the beginning of this exercise, 04_appendix.ipynb. And that code has already been run for you to save time, since the search code itself to perform this pre-processing can take upwards of 2 hours. For the time series, we'll use two different channels from the CSV file itself, corresponding to two different retrieval methods for estimating GPP from eddy covariance data. If you're interested, citations have been provided on the details of these two techniques. And here are the sources for how this GPP data is actually derived. So let's plot the time series now and see what it looks like. All right. We only have a little over 2 years worth of data to look at here, and that's enough for us to observe the cyclical nature of the data. Remember, both GPP and SIF are proxies for plant activity, so we would expect to see a seasonal pattern in the data. Indeed, we can see that the GPP peaks in the growing season from May to October, and that there are dormant seasons in winter and early spring from November through April. The deciduous broadleaf forest we are studying at this site exhibits rapid transitions during leaf out in spring and when the leaves fall in autumn. In this case, we can also note that the two GPP retrievals agree with each other pretty closely, represented by our two differently colored lines in the plot. Although there are some outliers where the two GPP retrieval methods do disagree, it's clear that the SIF data follows the same phenological trend of high values in the growing season and low values in the dormant season. Before we move on, let's think about the differences in the GPP and SIF data a little more critically. GPP is a measure of primary production and is therefore a direct measure of the carbon produced by plants through their photosynthesis. While SIF is a good proxy for photosynthesis activity, it only represents the light reactions in the plant. And when observed from space, only those plants representing the canopy, rather than the entire ecosystem, are included in the measurable SIF value that we get. Broadly speaking, observed SIF will depend on the light use efficiency of the plants being observed, which can vary by species and plant physiology. For example, a crop like corn, a common C4 photosynthesizer, has a different light use efficiency compared to soy, which uses C3 photosynthesis. Additionally, changes in plant health can have very fine spatial heterogeneity that affects the SIF signal in some places differently than in others. While a tower only observes a small area, the spatial aggregation technique that we use to get the SIF values that are included in this plot average samples across a broad area of several square kilometers. If there were an ecological disruption like a flood or a fire, how might you expect the SIF-GPP relationship that we're looking at here to be affected? Do you think that ecological disruptions can impact the phenological or seasonal patterns that we've noticed? At the end of the exercise, we'll use our second case study to run these plots again and see if there's any difference that we can note. Okay, so now that we've looked at the time series, let's try to correlate our remote sensing SIF data with the tower-based GPP data to see how related they are. You can see the general pattern of SIF follows the pattern of GPP in our plot, but we'd also like to quantify how well it does so. In this last cell in the section, we'll be performing that correlation. To do this, we'll create a scatter plot with SIF on the x-axis and GPP on the y-axis. For the GPP data, we'll choose the nighttime partitioned retrieval method from the tower, although you'll obtain a similar result using the daytime partition values, either of the lines on the plot. We'll add in a linear regression line using the sklearn module and compute an R-squared value. Finally, to make the points easier to parse, we'll give each point a color based on the season that it came from. We've provided the option to use different marker shapes depending on the year of the data point, but to keep the plot simple, we've disabled that feature by default. Let's run the code to create this scatter plot and see what we get. So here you can see that we've sorted data into seasons. We perform some simple filtering of our input data values. We create the plot here. and we calculate an R² value down here. So again, let's run the cell and see the result that we get. It's quite a lot of code, which I won't be going through in this exercise, but there's plenty of comments. So if you're interested in the specifics of the implementation, you can review it on your own after this exercise. And here we have the correlation plot. Our R² result of 0.73 suggests that these variables are well correlated, and it's only a little less than the R² value reported from comparing daily SIF to daily GPP in the Pierrat et al. 2022 paper that's the basis for this exercise. We can improve the correlation further by filtering the data on biome type when doing the pre-processing to create the JSON source file that we use for creating this plot in the first place. And that comes from the appendix, as I mentioned. As with the time series plot, we can see that there are some outlier points in the data, in particular these points over here. The points far to the right of the trend line suggest times when the observed SIF was higher than expected compared to the GPP value measured at the tower. We can use the SAM plot that we generated in section 1 to pull up context for one of these points so that we can observe if there's anything that we can notice about the spatial distribution of SIF points that could indicate why we get this result. The summer data point, this yellow one, furthest to the right corresponds to June 2nd, 2021, which we can note down by looking at the time series plot. Let's pull this up as a SAM plot. And we do so in the last cell over here. We can see that while there are some large SIF values near the tower indicated by the darker green, others are quite low, indicated by the light green or yellow. Additionally, there are gaps in the data caused by clouds and low-quality data. The SIF value we observed could have a variety of causes, such as our spatial aggregation picking up agricultural fields in the vicinity during peak growing season, or due to surface and isotropy playing a larger role at the time of the SAM acquisition. Unlike nadir mode, where the view zenith angle should be at or near 0, the SAM mode can have varying view zenith angle values between samples due to the action of the pointing mirror assembly. In this particular case, it looks like we have low data availability, noted by the lack of data up here, in the forested region directly around the tower, causing the cropland to the east, over here, to be overrepresented in the spatial average. We can test this observation by filtering our data in the appendix pre-processing step to only consider SIF samples with a deciduous forest or mixed forest biome classification. If you were to do that, you would see that the observed SIF at this outlier point would be closer to the trend line, and our R² would be improved to 0.76 overall. That's left as an exercise to the reader. So to review what we've learned so far and deepen our understanding of the SIF-GPP relationship, we'll move to the second case study of this exercise. By creating the plots in section 2 over again, we should hopefully see some different results. This time we'll be using the Metolius Forest site in Oregon, also called USME2. So I'll pull that up by searching for it directly in the Ameriflux website. I'll search for USME2. If you don't know the site identifier of your specific site, that's okay. You can find it by using the OCO3 SAM website that I mentioned at the beginning of this exercise. And here we see the second case study site. As opposed to the Michigan site, this location is an evergreen forest on the side of a mountain in the Cascade Range of the western U.S. Because we are talking about an evergreen forest now, one of the patterns you'll notice is a less pronounced drop-off and ramp-up in activity during seasonal transitions. Another important detail to note about USME2 is that there was a major wildfire in this region of Oregon in August 2020, and the area around the tower itself was burned. Despite this, the tower managed to continue collecting data, so we have a valuable resource on how this ecosystem responded to the disruption. Let's turn back over to the notebook and run the plots again to see what we can observe. So I'll scroll back up to the beginning of section 2, which is where we created the time series plot in the first cell of this section. And you can see we've already provided a line with the CSV file of Ameriflux data for the USME2 site, as well as our pre-processed SIF values from OCO3 that, again, come from the appendix notebook. I'll comment out the original lines that we used for producing our plots with UMBS site data, and I'll uncomment these lines to represent the USME2 site data, and then I'll run the cell again. We don't have a ton of pre-fire SIF and GPP data to work with, although we could look further back in the GPP record if we wanted to. We've cut it off to the beginning of the OCO3 mission for this particular exercise. This is still enough for us to see about 1 year worth of data before the fire and give us a picture of the phenological cycle in the Meteoroliths. This is enough for us to see about 1 year worth of data and gives us a picture of the phenological cycle in the Metolius forest region. As I mentioned, the dormant season down here is shorter and has less of a stark drop-off compared with the deciduous forest that we studied earlier. This can also partly be attributed to the climatology, although you would see similar effects when comparing forests with the same climate classification. And helpfully, these two sites have a very similar latitude. After the fire, GPP drops off significantly, noted by this flat line down here. Nevertheless, it is possible to see a small peak in activity during the summer of 2021, with a larger peak, at least in the nighttime partitioned data, in 2022. This recovery around the tower can likely be attributed to cirral growth in the region, where the burn scars filled in with grasses and opportunistic brush. The cirral growth is stronger in the subsequent year, possibly due to young trees taking the place of burned and logged mature trees affected by the fire. Now let's look at the SIF values that we see in this plot. While the initial SIF values from 2019 and 2020 agree with the GPP behavior around the tower, the two measurements diverge after the fire. In summer 2021 and 2022, we see peaks in SIF to a similar level that we saw prior to the fire, a trend that is not reflected in GPP. When the wildfire burned through this region, not every area was as heavily impacted as the region directly around the tower site. Therefore, we can still observe some SIF activity from unaffected strands of trees in the vicinity. Now let's generate our regression plot for this example and see what information we can note. And again, as with the first cell that we ran, I'm going to uncomment these two lines containing the updated GPP and SIF file. Okay. Our R-squared value is significantly worse than the Michigan site at just 0.121. In the absence of additional context, this might suggest that these variables are almost uncorrelated. But as we observed in the time series, there's still a seasonal pattern to SIF. It just diverges from the GPP data due to the effects of the fire. Taking a look at the SAM plot for a specific date can help to make this more apparent. For the final plot of this exercise, let's pull up a SAM of the Metolius forest region before and after the fire to see what spatial patterns we can observe, at least qualitatively. For the pre-fire data point, we can look at June 19th, 2020, and then after the fire, we can study May 28th, 2021. We can plot those two SAMs ourselves using the notebook. However, you'll have to find the SAM extents manually by checking against the official OCO3 plots. An easier way for us to do this is to just look at the official OCO3 SAM plots themselves, since they've already been generated for us. So I'll return to the OCO3 SAM website and I'll find the US ME2 site on the map. I know it's in the US state of Oregon, so I'll zoom in. And the SIF low site that we can see here is referred to as EcoStress US ME2 on the map. Let's now search for all of the SAM acquisitions. The first date we'll want to use is June 19th, 2020, as I mentioned. And let's pull up the tab with the plots. If I scroll down to the SIF 757 nanometer plot, we can take a closer look at the spatial features that we note. In the 2020 data, we note a pattern of growing season behavior in this region under normal conditions. SIF is generally higher in this season, despite a somewhat lower average value compared with the GPP, but we can observe that the area around the tower has a somewhat lower value and low data availability. It's only when we average points from around the region of the tower that we achieve the value that you can see in the time series and the correlation plot. Now let's take a look at the 2021 data to see if we can note anything different about that particular acquisition. So I'll go to the next page of results, and I'll go to May 28th. And let me pull up this plot. In this example, we noted a much higher SIF value than the corresponding GPP value in our time series. Let me turn back over to the time series plot so that you can see what I'm talking about. Over here, you can see the time series for this site. And we're looking at May 2021, which is around here. And SIF is peaking for the growing season in this region, despite GPP being very low around the tower site. Let's turn back over to the plot and see what we can notice. Data availability is still quite sparse, like we saw in 2020, around the tower site. But in the 2021 data, we can definitely see that the value closest to the tower is relatively low compared to the surrounding area. There are sporadic samples in the forest in this region that have high SIF, these darker green values, especially higher up in the mountain range, that show that the spatial heterogeneity of this region is quite high. This suggests that the fire affected different regions of this forest in quite a fine pattern, with areas that were completely burned in some regions and unaffected in other areas. You can investigate this relationship by analyzing more dates and even other sites on your own. And now that we've explained the basics of performing a SIF-GPP correlation, you should be able to perform it for any arbitrary site within the Ameriflux dataset. That concludes this exercise, and thank you very much for your attention.
Thank you very much, Jackie, for that great demonstration, and to Dr. Liu for the great presentation. Next, I will provide a summary of today's session. So in summary, OCO2 has global coverage, while OCO3 has observations between more or less 52 degrees north-south latitude and at different times of day. OCO3 has an extra observing mode, which is called the SAM mode, the snapshot area mode. And level 2 and level 3 SIF data can be used for understanding or addressing different applications, such as understanding the impact of climate perturbations on vegetation health, drought monitoring, and crop yield prediction. And here's a list of resources for those wishing to dive deeper into the material that was presented today. If you have any questions about the material presented today, please feel free to contact Dr. Junjie Liu, Jackie Ryan, or Karen Yuan. And then just a reminder that there is a third and final session next week, next Wednesday, October 29th at the same time. And that session will run Jupyter Notebooks to compare how gap-filled SIF products such as GoSIF can be used for assessing the impact of floods and droughts on cropland through specific case studies in the Midwest. You'll be using Jupyter Notebook tutorials to choose another region or time frame of your choice and reproduce new products for another analysis. And you'll compare SIF products aggregated in space and time using open source tools and how they can be used to study vegetation change across different regions in a variety of science and applied use cases. So with that, we've reached the end now of session 2. Thank you again to our guest instructors and to the participants for their interest, and we've been receiving a lot of questions. So we will be starting the Q&A session now. So we've been compiling your questions onto a document, and we will go through the questions, and we will also edit this document and post it on the training page within a couple of days. All right, so let's just start from the top down. The first question, will snow and oceans show as black? Could we still see pine trees, or will it appear black due to snow? So Dr. Liu, if you'd like to go ahead and respond to that. So her response is that snow is highly absorbing at the near infrared band. So it does appear black, and snow can reduce the amount of light reaching the canopy and as a result reduce the SIF signal. That said, SIF signals from non-dormant photosystems are detected in evergreen needle leaf forests during winter and spring in the presence of snow. And she has included here a reference article if you would like to dig deeper into this. Great. Let's go on to the second question. I'm doing a SIF study for different biomes in India. Where can I get Flux Tower GPP data for different biomes to compare with SIF-generated GPP values? And her response is that you can find the Flux Tower data at the FluxNet website with the link that is posted here. However, the coverage in India is somewhat limited, and you will want to check whether India has Flux Tower data that is not part of FluxNet. So there are many Flux Towers around the world. Obviously, there's the FluxNet network, but there might be other networks, and you might want to check. Let's go on with the next question. Correlation between SIF and GPP tower data within each season does not appear to be strong other than in the fall. This seems to suggest that we cannot reliably interpret changes within a season, even in the summer, outside the noise. So I will let maybe either Dr. Liu, if she has resolved her technical issues, or Jackie respond to that.
I do think that's a little bit of a better question for Dr. Liu, but I'll try to answer it as best as I can.
Right.
So towards the end of the notebook, I include some suggestions on how to improve the correlation. One of the challenges that we have with using SAM mode data is, as I mentioned, like data can be very sparse around a tower for any given observation, which requires us to do the spatial aggregation. But at the same time, because of that, we're including a lot of points that aren't necessarily the same as the tower site itself. So typically the best comparison that you'll have is when a tower has photospec as well, which is relatively uncommon. With that being said, you can still use the data, but it is, like I mentioned, recommended to filter by biome type.
Okay, thank you very much for that response. We'll go on to question number 4. What is the difference between daily SIF 740 nanometers and SIF 740 nanometers? So I'll go ahead and read the response that's being typed. Daily SIF is the daily mean SIF, so it's the average value, which accounts for changes in solar radiance throughout the day, while SIF 740, so daily SIF 740 is the average value, while SIF 740 is the instantaneous SIF value retrieved from that specific band, from that specific 740 nanometer band. The next question, question number 5. What are the specific characteristics of each retrieval band? Is there a preference between 757 nanometers over 740 nanometers?
I'll let Junjie add any details if she'd like, but the 740 nanometer data will generally have a stronger SIF signal, but it doesn't come from a measurement taken directly by the instrument. So it's interpolated from the 757 and 771 data. If you would prefer to use a measurement directly from the instrument, that's a case when you would want to use 757 instead.
Okay, thank you. And let's move on. So it looks like that's the last question. However, I believe there have been new questions added. Let me take a look. No, it looks like that's the last question. Any additional questions, please go ahead and write them. Or what we'll do is any additional questions that we receive. Oh, here's one. Okay. So let me just include it in the, yeah. All right. So question number 6, why do the resulting data visualizations appear as sparse pixels that don't cover the entire study area? So assuming we define the specific geographical boundaries, what methods are used to compensate for the missing data gaps in the final map products?
Generally speaking, you can use gap-filled products if the data availability is not enough for your use case. Other than that, there's not really a way for us to improve upon the gaps without synthesizing or estimating data from other predictors. So with SAM, the gaps occur due to bad data points or the presence of heavy clouds, for instance, preventing the estimate.
Okay, thank you very much, Jackie. The next question, regarding historical baseline data for regional studies, since satellite imagery isn't available for periods like the early 1900s, could you recommend any key repositories for declassified aerial photography or historical topographic maps that are freely accessible for scientific use?
I'm sorry, I don't know the answer to that question.
Yeah, I think we'll have to take a look unless Karen or Yunji, if you have any insight into this. Otherwise, we will look into it and respond it in the document.
Yeah, I think this is dependent certainly on the area you want to look at, the country you want to look at, because obviously aerial photography and the practice of it would be much later on. When you're saying the early 1900s, that would be difficult to get anything in the 1900s just because of the availability of the equipment to be able to do that and to do the surveillance. I would be hard pressed to find anything before 1950, I think. And also the historical topographic maps, I would have to check when USGS or other agencies would have started collecting that, because that would all be produced during land surveys. So that would also be country dependent.
Thank you very much, Karen. Okay, we have another question or more of a state. Okay, so it says the measurements have been directly or have the measurements been directly acquired from sensors? I'm not sure if this is a question or it says the lesson seems more advanced now.
So, yes, what we were doing in this lesson was comparing two different sensors. One was from space and one was on the ground. The continuous line plot that we looked at in the time series was the ground-based data. And that's a real sensor. The SAM plots also were real acquisitions taken over what we can consider as like one-time points from the OCO3 instrument.
All right, so I think that is it in terms of questions. Just to remind you, there is one more session as part of this training series, and that session will be next Wednesday at the same time. We're going to be looking at gap-filled products. It's going to be primarily a demonstration that Jackie will be leading. So please tune in. And as a reminder about the homework, there is one homework associated with this training, and that will open up next Wednesday, and it'll be posted on the training webpage. All right. With that, we've then reached the end of the second session. I'd like to give the opportunity to our guest experts to say some final words. So, Jackie, if you would like to say some words before we close.
Yeah, I have two comments. There was actually another question that came in. Is it possible to integrate a second variable such as land surface temperature or soil moisture with the SIF data to understand the underlying changes or causes of the observed changes in vegetation health? And this is an area of active research within our science community. And on the OCO3 website, there's a recently released product that combines EcoStress with OCO3. So EcoStress provides land surface temperature, and it enables you to calculate water use efficiency. So if we do eventually do an advanced course, that might be a topic of discussion that we would cover there.
Yeah, absolutely. Thanks for bringing that up. And I think data integration is always a great interest in for our ARSET community. So certainly something to consider. Go ahead.
The second comment that I wanted to make was a lot of you last time mentioned that you use Conda for your Python environment. And so I added a Conda setup script for both Mac and Windows. So you should be able to install the course materials using Conda now.
Thanks, Jackie. And Karen, would you like to say some closing words?
Just thank you to all the participants for joining us today. And please join us next week because I think for those who want to work with our data, you'd be very pleased with what Jackie has put together. And as you can see, she's very responsive, real time, and follow up for to help and support our users. So we look forward to seeing you next week as well. Thank you.
Thank you very much, Karen. Again, thank you to our invited experts, to Jackie, Junji, and Karen for today's presentation demonstration. And of course, thank you to all of the participants for tuning in, for your interest, for your questions and enthusiasm about this topic. And finally, I'd like to thank the ARSET team for making this possible. So with that, we will see each other in one more week. So until then, I'm wishing you all a good day. Bye-bye.